right. Well, thank you everyone for joining us today. I want to welcome everyone into our webinar. It is called Know Before You Grow, Microbial Contract Manufacturing with MDG. My name is Judd Reminger. I will be the moderator today. As a lot of you come into our chat room, please utilize our chat feature, which is on the right-hand side of your screen. I know we're going to be going through a lot of stuff today in the webinar, but if you have any questions or observations or want to talk during the presentation, please use that chat feature on your right-hand side of the screen. Also, we will be having after the webinar email that will go out where you have this entire webinar that will be seen for you to watch back, and also a short survey that we will have for the after webinar, and then also any other questions that you might have stemming from today. So our speakers that will be joining us today, our speakers is Shannon Lousing. She is a Director of Quality and Regulatory, and she has over 10 years experience here at MDG in food/feed safety and vendor ingredient approval. We also have Stephanie Weber, her Research Manager of Process Development. She has 10 years of experience here at MDG in strain optimization and media processing development. So without further ado, take it away.
You can go to the next slide, Judd.
Good afternoon. Helps if I take the speaker off of mute before I start talking. So, yeah, where are you right now? That's one of the first questions that we ask when we get to meet you. If you come in for a visit, or if we're going to have a video call or a conference call about what your goals are, what you would like to achieve to get to learn about you, your company, and the organism that you have. So we ask you, what's your manufacturing goal, what's your scope of work, and what stage of development you're at. One of the first things we do to see if you are a fit by asking those questions is your organism biosafety level 1? We only grow biosafety level 1s. Growing anything higher than that takes a special permit, and that's not the focus of our manufacturing. We are primary bacillus, and anything that's just safe to grow without extra containment is a biosafety level 1. We also ask, is your organism genetically modified? And what we're looking for is, does your organism have any foreign material that you've inserted into it? At this time, we're not to the point of being able to accept a GMO, one that has that foreign DNA inserted into it, into our facility. We're not quite there yet. If you've done some gene editing on your organism, that is an organism that is a fit. So that would be one of the organisms that is a fit. And then, how aerobic is your organism? Since bacillus growers, we use a lot of stir and a lot of air. So how much air does your organism need? If it's full anaerobe, that's probably not a fit at this time. If it needs some air, well, that would be something we would find out in deeper discussion if the needs that you have or what your strain needs is a fit for our equipment, but that goes on to the next step. So that's a maybe a fit. And if you're an aerobic organism, you are definitely a fit for our processing and our equipment. And then we'd ask next, are you batch fed? So we're not quite there with batch fed yet. It's something we're working on, but we do have the ability to add filtered ingredients. So that tells us if you're fit or not. If each of those is a yes, I'm a BSL1. No, I do not have foreign material inserted into my microorganism. Hey, I'm aerobic or mostly aerobic for the growth of the organism and don't need batch fed. Then you were definitely a match, and we would continue the conversation and find out some more. What are your manufacturing goals? Are you looking for process development, help scaling all the way up? Do you just need some spray drying? We do some pilot spray drying. Or are you ready to go to commercial scale? So what is your goal and what stage of development are you at? Very closely tied together in your goal. Are you looking for just the biomass production? So we grow the microorganism, separate it from the media, dry it, and send you back the culture. Are you looking for the fermentation broth collection or the growth broth? And you want us to throw away the microorganism, but you would like us to collect the broth and send that back. You know, that's another thing that is a fit. Collection, if you want everything sent back as liquid, we can do that too. And then what's your drying format? That's another thing that we discuss. Stephanie, anything else at this stage before we get to your group?
I think the only other thing to mention would be, like you had said, what stage of development are you in? So have you already done some pilot studies on your end where you have potential parameters or media formulations that you know work for your strain? During those conversations, we like to go back and forth with that as well so that we can really do more of like a copy paste to our fermentation tank. So maybe you don't necessarily need true optimization through the entire course of process development, but process development can help support you in transferring some of that information that you would have about your strain to our commercial side.
Slide.
Yeah, so Shannon touched on most of this in the first slide. Again, if an organism's growth process aerobic or anaerobic, we do mainly work with bacillus, so those are highly aerobic. So both in lab process development and on our fermentation side, we aren't quite there for anaerobic growth yet. We mainly do batch fermentation. But as Shannon mentioned, we are able to do filter sterilization, both in lab and again on the fermentation scale as well.
And then on the regulatory side, MDG has, we manufacture a number of food and feed streams, food for people, food for animals. So what kind of regulatory do you need? Are you looking for a direct fed microbial, that animal feed strain? Is it a food ingredient strain, goes into beverage food, maybe you're going to use it in a dietary supplement, or is it for an industrial application? Each of those paths has some different requirements that we use in guidelines that we have to go by to meet the regulatory compliance for food and feed. So that's another thing that we ask, are we a fit for that? You may have some additional requirements that we'd have to discuss, especially on the food and feed side. How can we work that into the schedule and into the needs? How do we provide information for you to do your registration? Because we're going to assume that you're going to be doing sales maybe outside of the US, or you're going to go for your GRAS with the FDA, and then how do we support you that way? And we do that early on in the talks in the Are We a Fit portion of the sales cycle and the development cycle. Slide, Steph?
Yeah, so we're a fit. So next question, obviously, what's next? This is really kind of where process development comes into the custom fermentation process. So usually, as Shannon mentioned, a lot of those Are We a Fit conversations have happened. NDAs potentially have been signed. Things are moving forward. Initially, usually process development and the customer at this point will have one of those initial meetings. That's where we like to understand where you are in your process Do you already have a formulation and tank parameters that you can share that we can test either in lab or help to formulate transfer documents so that we can put it out on our commercial floor? Now, if you are looking for optimization, we do work in kind of a 3-phase format. So usually phase 1 is the strain work. That is getting a strain from you, the customer, brought into MDG. And what we do during our phase 1 is we're checking purity of that source plate that was sent to us by you. So what we're looking for is making sure no contaminants have found their way onto the plate. We generate a DNA fingerprint with our in-house methods and look to verify that that is all one strain. Once we have verified that it is one strain, we will use your source plate to generate our own cryo vials or working cell banks. And through that process, in the end, we generate another streak plate, which we send out back to you, the customer, to verify what we have made in-house is what you're expecting. So basically the strain you sent us is the strain you get back. So that's really that phase 1. At that point, we do usually wait to hear feedback from the customer to ensure that they give us the go-ahead that yes, what we got back from you is exactly what we were expecting. Please move ahead, and we'll go into that phase 2. So phase 2 is usually about 4 to 6 weeks. That's more on our small scale. So that we utilize microtiter plates. We can look at shake flasks. We can do even testing in some of our 1.4 total volume bioreactors. So we do have some smaller bioreactors that we can do testing in as well during phase 2. And really that's where we're identifying some of the nutrient factors that may be needed for your strain. So looking at up to 200 different sources at one time on our microtiter plates. And then we can really scale that down to generating maybe a couple few media formulations. Obviously looking for, if you're looking for best spore titer or biomass yield, that's what we would use as a benchmark during our phase 2 testing. Once we have a good idea of a few media formulations, we will then go into our phase 3. There, we usually like to spend about 6 weeks. Those are in the 5-liter benchtop fermenters. Here we are able to do a little bit more control, looking at our pHs, our dissolved oxygen, our aeration, our agitation, really looking to scale down from our big fermenters to labs so that we get the best idea of what to expect when it goes to the commercial floor. At the end of phase 3, we give back a pilot powder from our benchtop freeze dryer. And that's really so that the customer or you can verify that the product maybe meets a stability requirement that you might have, or maybe it's an enumeration. You also want to verify the count that you're getting from our testing as well. And that kind of wraps up.
Stephanie.
Yeah.
Oh, I'm going to say, have we had any customers that come to us with a cell bank already? How would that shift your process, or does it not impact the length of the phase 2 or phase 1?
Yeah, that's a great question, Shannon. We have had customers who do like their own working cell banks. They verify their purity, they're comfortable with what they have, and they will send those to us. Usually that takes care of the phase 1 work. So that'll shave off, probably 2 to 3 weeks of work, and we can jump straight into both of those optimization phases then at that point.
What if they came with a formula that they've already been using? Maybe they have had success in their own pilot. How does that impact the stage 2 and stage 3?
Yeah, so usually we like to look, maybe there's some media pieces that we actually utilize in-house as well. Usually that works out well in speeding up the process, but we have had to do kind of a parallel process where we do have to go and source potential ingredients that you may have in your formulations. That's where then process development will work kind of hand in hand with Shannon to get vendor approvals, looking at if there's any regulatory when it comes to, do you have a feed strain, a food strain? So there's some parallel work that can be done there to make sure that, we stay within that process and that timeline.
You talk about the food and feed requirements. So one of the things for food strains that we do here is we don't use allergen ingredients and we don't use any animal-derived ingredients. So the media recipe is pretty simple, and it's do that on purpose, the excluding allergens and the animal-derived ingredients to make it much more simple for you to be able to sell your product. That way you can go for kosher, you can go for a halal, like non-GMO, because we even work with sourcing certified organic or non-GMO ingredients, to make sure it's not from a GMO plant. When you're using veggie-derived materials, that's one of the things that we have, and it makes the process very nice, at least from the regulatory perspective, is that there's not all the declarations that you need for end user use with that allergen-free and animal derived ingredient-free media recipe. But I know it does give Stephanie's group, they have to work a little bit harder on the process parameters to make that media formula work its best. Anything else? Are we ready for the next slide?
I think we're ready for next slide. Yeah, so now
and there's your parallel
process. Yeah, so while like we had just talked about the supplier approval for food or feed, so while process development is in the optimization, there's a lot of parallel processes or conversations that are happening to ensure that we meet, your potential manufacturing goals and timelines. Not only looking at approving ingredients, food or feed, there's also internal, working with quality control, making sure that they're aware of your specifications, maybe morphologies, things to expect once it goes to commercial and through to that quality check. Also, parallel is a drying process. So are you interested in potential spray drying or freeze drying, where we can look at, different ingredients or carriers or things that are needed in your process, can all be looked at all within that same optimization timeline. So we try to keep all the conversations going so that we can get to commercial as fast as needed for your strain.
One of the things we do during one of our parallel processes is aligning customer QC with MDG QC, and that comes down to the enumeration. In our QC lab, we do have a method for bacillus that we use, and we proficiency test on that. So it's one that we always like to do when we're doing our QC. And you have your own methodology. So we get together and test sample, each using our own method, and see how closely we come together. And then we look at, as a group, where we could fine tune the methodology for your organism. So that's one of the items we do in parallel. Supplier approval. If you're a food and feed person, you need to make us an approved supplier. So we're doing the food safety at the same time. You're going through setting up the customer audit. So you're able to send your QA team in, your food safety team, they do the on-site audit, because that's quite a bit to get done in a 12-week time frame as well. Is there maybe some other registration work that needs to be done? Because maybe you're exporting out of the country. That's something else we try to work on in parallel. And then drying, that's a process, and maybe we're looking at drying parameters as well. And we run a larger scale run with material that you've already grown, because our pilot spray dryer takes, what is it, 5 liters, Stephanie?
I think
5 liters.
gallon.
Oh, gallon. That's nice. That's even less than 5 liters. I think if you're looking at spray drying and you have material that you've been able to grow, process development can work on the growth parameters and getting to commercial scale here in our equipment, while our spray drying team can work on the spray drying parameters at the same time in the P10 while we're doing the QC enumeration alignment and any supplier approval work that has to be done, those questionnaires and the on-site audits.
And I should correct myself. I believe I think it's 20 gallons. Sorry, let me correct myself.
Oh.
Because unfortunately, we don't generate enough material in our 5 liters to be able to do any P10 pilot spray drying. So usually, like Shannon mentioned, if you do have some material we can experiment with and optimize on our P10. Otherwise, there are instances where we can take from the first commercial run and look at drying optimization from that as well.
Very dry. Is there anything that we haven't covered yet? I'm trying to think about. We've got our in parallel. We talked about enumeration because that's always the fun one, aligning labs. And fun thing is that we do it over video like this. One of the QC team she really enjoys training, and she does like a live demo, and it's set up like a cooking show. And she does it over video like this, and she has all the materials, and she walks through the method and explains key portions of the SOP. I think that's always fun. I get to attend those with her, and I enjoy that actually a lot because it's a nice way to meet the QC team for the customer, for you, and get to know them because we will be talking in the future, and it's good to establish that relationship. That's probably one of my favorite parts. Enjoy that quite a lot. And then when you already mentioned about sending all the information to the QC team when we're ready to go to commercial, all that, we get internal tech transfer as well, so that we have our QC parameters set up for testing. We do that positive release, so everything has to meet the specification, colony morphology, the DNA fingerprint, if there's a minimum colony forming unit count, maybe there's a moisture or a water activity that you send to us so we can get it set up in our inventory and LIMS system.
Yeah, and I think at any point during this optimization process, going through some of these parallel processes, we do keep you informed on a pretty regular basis. The process development team likes to give updates at least bi-weekly. If we do run into any issue where maybe something's not growing as well as we had hoped, we do communicate. We jump on Teams calls. Maybe your technical team and the process development team can really get together and help drive it forward quicker as well. So there is a lot of communication with you as well during this process so that you know where we are at each stage. Yeah, I can't think of anything else at this point.
Judd, I think that's the end of what we have to share.
Well, thank you guys very much for your wealth of knowledge. And if you have any questions, please, you can put them in the chat right now. And we will, after this webinar, email you separately from our experts and really dive deep into your question. As well as be on the lookout through your email. We will have a survey that you can go to, and I just put that in the chat as well, and we'll just tell you how we did. And then also, if you have any other follow-ups, we can talk to you through that. But I want to thank Steph, Shannon. I want to thank both of you. You guys did a great job, and I know a lot of people that were watching and really, really loved all the info. So thank everyone for tuning in. And again, if you have any questions, please put them in the chat, or you can email us at info@mdgbio.com. Once again, thank our lovely speakers, and we will see you guys next time for an MDU webinar.
Thank you.
Thank you.